Hello friends, today we'll discuss about cytoskeleton. Starting with what is cytoskeleton? It is intracellular complex network of fibers. How it works? It maintains structural integrity of the cell. It allows the cell to change the shape as well as it allows the cell to move. It also helps to participate cell in various physiological activities. Elements of the cytoskeleton, they are made up of different cell proteins and they consist of microtubules, intermediate filaments and microfilaments. Starting with microtubules, these are long hollow tubular structure you can see here in the diagram. These are the structure without limiting membrane. They are about 25 nanometer in diameter. And these microtubules are made up of tubulin dimer you can see in the diagram. It is made up of two subunits of globular protein. They are alpha and beta tubulin. Function of tubulin bundle. This tubulin bundle gives structural strength to the cell. Now, functions of microtubule. Microtubules are important transport systems of the cell. Some organelles like chromosomes, mitochondria, secretory vesicles and protein molecules, they move from one part of the cell to other part of the cell through microtubules. Microtubules also contribute to the maintenance of cell strength as well as the shape of the cell. They also help in the formation of spindle. You can see here at the time of cell division, spindles they are formed. And these spindles they help to move the chromosomes during mitotic cell division. Microtubules are also pillars for the structure and functions of the cilia. Microtubules they also contribute to cell motility. Molecular motors, they are kinesin and dynein. They help in the movement of molecules through these microtubules. You can see here, these are the molecular motor, this one, and they help in the movement of various molecules. One of the head of the molecular motor, this is kinesin molecular motor, one head is bound to the microtubule. Now, cilia and flagella. What is cilia and flagella? Cilia are the hair-like projections from the cell and flagella, that is a lash-like appendage that helps the movement. The cilia and flagella, they both are made up of microtubules enclosed in the plasma and they are active in the locomotion of the cell. You can see here this is the structure of the cilia and flagella containing microtubules. Now, applied physiology. Tubulin subunits, alpha and beta tubulin as we have discussed, they have property of disaggregation which is also known as disassembly and reaggregation which is also known as assembly. This process of disassembly and assembly they occur simultaneously. Many drugs they act by inhibiting the microtubule assembly. Growth of the microtubule is also sensitive to temperature, like cold condition favors disassembly of the microtubule. As well as interaction with GTP, you can see here, it facilitates the formation of microtubule. Next one, that is intermediate filaments. You can see here, intermediate filaments, they are filamentous structure, about 8 to 14 nanometer in diameter. Some filaments, they connect nuclear membrane to the cell membrane, you can see here in the diagram. Functions of intermediate filaments. As they connect nuclear membrane to the cell membrane as well as the membrane of the cell organelle. So, it mechanically integrates cell organelle with the cytoplasm. Also, it is additional skeletal network for cell and also it resists rupture of the cell when external pressure is applied. Applied physiology, when these intermediate filaments they are absent, it results in easy rupture of the cell. And if these intermediate filaments they are abnormal, blistering of skin is common. These intermediate filaments are also used as cell marker. How? Proteins present on the intermediate filaments, they are cell specific and therefore they are used as cell marker. For example, cytoskeleton, that is a cell marker protein present on the intermediate filament of the epithelial cell, and vimentin, that is the cell marker protein present on the fibroblast. Next is microfilaments. These are long solid filamentous structure, diameter of about 4 to 8 nanometer, and they are made up of contractile proteins. They are actin and myosin. Actin is the most abundant protein in the mammalian cell. You can see here, as you all know, actin is present in the globular form, that is G-actin, and this G-actin is polymerized to form filamentous actin, which is known as F-actin. This actin attaches to various parts of the cytoskeleton by other protein. These other proteins are known as anchoring proteins and they are identified by number like 4.1, 4.2 and 4.9. Special characteristics of microfilaments. These microfilaments are well developed in muscle cell, but they are also present in all the cells and they are better organized in the cells. They secrete the granules, and the granular secretion of the cell, that is by exocytosis. In those cells, these microfilaments are well developed. They are also important for the platelets to change their shape, as well as they also help to move the granules of the platelets, and these granules are moved from inside of the platelet to the canaliculi of the platelet, and therefore these granules, they can release chemicals easily. Also, they are present in the cells with the microvilli. Microvilli are present on the epithelial surface. If microvilli, you can see they are present. This microvilli structure contains microfilaments. So they are also important for the functions of microvilli. So now functions of microfilaments. Number one, they are the major contractile fibers and therefore they help in the change of the shape of the cell, as well as they are important for the movement of the cell. Second function, they have contractile mechanism, contractile phenomena, and this contractile phenomena is important for phagocytosis, also for the transport, also for the secretion. Cell contents are secreted, also important for the contraction of the cell, discharge of the contents, and also they are important for the movements of the secretory granules. As we have discussed, microfilaments, they are present in the microvilli. So they also help in the function of microvilli. What is the function? That is absorptive surface of the cell is increased by the microvilli, and that is enhanced by the microfilaments. In skeletal muscles, actin and myosin filaments which are present, that is also important for the contractile property of the skeletal muscle. So now functions of cytoskeleton, that is we are enumerating them. Here in the diagram you can see one, that is it is important for the structural strength, it provides support. It is also important for the intracellular transport of the substances. It is also important for the contractility of the cell, important for the motility and also for the spatial organization. So this is all about cytoskeleton. Thank you.